So I'm going to talk a bit about how you analyze cancer sequencing data, and in particular, I will do it from the perspective of an embedded bioinformatician. So I'm Chris, and I am part of Majewski lab that's working with cancer, and particular focus on DNA repair and cancer drug resistance. So we have our research questions, and to answer them, we generate different kinds of data, and the data I deal with is mostly exomes, genomes, and RNA sequencing, and this is bulk sequencing. We also collaborate quite a lot, both within WEHI and externally as well, and the collaborators will come with their own research questions and their own data, and all of this data, both from the lab and from collaborators, then end up with me. And we also use public data sometimes from TCGA, Cosmic, and that can be different kinds of resources, but can also be more RNA-Seq data, exomes, or genomes. With all this information, the typical analysis cycle is to quality control the samples, make sure to the extent we can that samples are okay. We then analyze the samples, and what I mean with analysis here is like the actual variant calling, the actual copy number calling, like the statsy, mathy part of the analysis. And we then use the output of that to try as best as we can to answer the research questions that we or the collaborators are looking for. And I then try to report back to the research question, either within the lab or to collaborators. And I've been an embedded bioinformatician for many years, and we've gone through this cycle, I think, close to 100 batches, and we're getting pretty good at this part here. So what we've been doing over the years is that we've been collecting all of these different steps into our package that's called SuperFreq, and that's available on GitHub and so on. And this package is helping you with all of these 3 steps. It's doing the somatic SNP calling, the copy number calling, as well as the clonal tracking. And this is a little bit different from a normal method that's usually doing like one specific part of the analysis, right? You have a specific one that's doing the copy number calling, or a specific one that's doing the variant calling. Well, here we are aiming to help you go through this entire part of the quality control and the research question, which involves things like more visualization and diagnostics for the quality control. And we also do quality of life things like help you annotate the somatic variants and so on to make it easier to answer the research questions. So while in programming and bioinformatics in general, you will often see the Linux main philosophy, which is do one thing, do it well. That has not been the guiding principle for us, but it's partially a matter of quality of life for the user that needs everything done, not have to use a lot of different methods. And it's also good that the different parts can inform each other. It's a lot of crosstalk between the different analyses. That's quite difficult to do if they're done with different methods. Since we sometimes have precious samples, both from us and from collaborators, we can't always afford a strict garbage in, garbage out strategy, which means that if the data doesn't meet a certain quality threshold, you just ignore it, say, give me better data, I'm not going to analyze it. Instead, we've put quite a lot of effort into salvage what little information is there, even in noisier samples, and try to adapt the statistics and the algorithm to be able to cope with a little bit more variance, a little bit more bias. So this also departs a little bit from the standard of assessing methods that tend to go to very clean samples, simulated data, and so on, with very little biases, very little noise, and then try to maximize sensitivity as much as possible on that kind of data, bringing sensitivity from like 95 to 96% and so on. This also has not been our guiding principles, but I'm not saying it's bad. We obviously want as high sensitivity as possible, but we've also looked a lot at real data. We've been trying really hard to test our methods on real data and not always super clean data to make sure that we not only have the sensitivity, but we also manage false positives within reasonable levels. So the SuperFreq workflow consists of samples from the individual So we're looking at the BAM files. The BAM files are the input. So it's the individual that we are analyzing, and we also require reference normal samples. So these are unrelated normal samples, but should be from the same technology, and ideally should be like from the same lab. The more similar the reference samples are to the study samples, technically, in terms of like generated by the same people and so on. The closer it is, the better. We then call somatic variants, and together with the germline variants and gene counts, we also call copy numbers. And we then use the somatic variants and the copy number calls to track clones and phylogeny. And this talk, I'm going to go through the different parts and talk a bit about how the guiding principles has influenced different parts of it, and show some examples of data that we've come across. So I'm going to start in the middle and talk about the copy number alterations. And I'm in particular going to talk about how we can run this on RNA sequencing, which is not a standard analysis. In general, when you call copy numbers from sequencing data, you look at two things. You look at the read depth, and you look at the frequency of the B allele of the heterozygous germline SNPs. So this is a cartoon of a cell here with a maternal and a paternal allele, and each one has its own unique variants. These are the heterozygous SNPs that are present on only one of the two alleles. So we will have a certain read depth in the sequencing data, and both of these alleles, either on the A or the B allele, will come in at about 50% B allele frequency. However, if we have copy number changes, for example, if we have a second copy of A, we're going to have 3 copies, and we're going to have 50% more coverage over this area. And also the 50% B allele frequencies are going to shift to 67 or 33, depending on if the variant is on the gained or the other allele. And a similar situation, if we lose a copy, we're going to see a decrease in read depth, and we're going to see even more shifting B allele frequencies. For a genome, this data is very clean. So this is the read depth log fold change. We compare this to the reference normal. So we see a log fold change of 0 here, means that we have the same read depth as in the reference normals, which means that we have 2 copies of DNA. And we see the B allele frequencies here that we run between 0 and 0.5, and allele frequencies larger than 0.5 have just been mirrored down to the lower part instead. So we're only looking at the smaller frequency. And we see here that this data is quite clean. Like, it's really easy to see from far away what's going on here. And it's just looking at this data, it seems quite easy to write an algorithm that calls copy numbers from genome data. And there are plenty of algorithms that do that as well. Looking at exome, we see a little bit more variance, a little bit more spread out here, but there's still like a decent signal to noise ratio here. We see a gain here, loss here, see the signal in the B allele frequencies and so on. And there are lots and lots of methods that call copy numbers from exomes. I think we have a list of at least 30 or something like that. If you go to RNA-Seq data, you are looking at these kind of things. So here, the log fold change is influenced by the expression. So it's not only the number of copies of DNA, but highly expressed genes will have a much higher read depth than not expressed genes. So this is giving rise to a huge variability in the log fold change here. Variant calling is also a little bit more difficult in RNA-Seq, and we see a bit more variability here, although we still see a little bit of signal here at chromosome 7. So I'm going to talk a bit about the workflow for calling RNA-Seq, and in particular, the one change that we had to make to get it working on RNA-Seq. So this is test sample that we're trying to call on, and we're comparing to the reference samples. And we then call variance to get the B allele frequencies. And I'm going to focus more on this part here, how we get the log fold change and look at the read depth. Because the first step here is to do the read counts. And we then want to compare the read counts in the test sample compared to the reference samples. And this is essentially a differential expression analysis, right? This is comparing counts against counts. And this is something that a lot of people, many people in the audience would have done differential expression analysis and know all the things here. And I'm going to reuse a lot of that machinery. In particular, I'm going to use Luma. And and VUM to study the differences here. But there are also a few other tools that are available from the structure of the copy number alterations that we can benefit from. So the first thing we do after getting the counts is that we correct for GC bias. So this is a quite basic thing. We see the GC content here, and in this particular sample, we see that there's a slight drop in read-depth. This is log 2 read-depth on the y-axis here. We see a slight drop for high GC content, and this is related to the PCR settings and a lot of other things going on in the lab. So we just fit the low S curve to this one, and then we correct for it essentially by dividing each gene by the low S fit of the local GC content. And this is done individually for each sample and each reference sample. Once we have these pseudo-counts afterwards, we can't really use ZR with pseudo-counts. You kind of want real counts for that. So instead, we go to CPMs and we use Lemma and VUM. So many of you will have seen this curve here on the x-axis. It's essentially the expression of the gene. And we see that the genes kind of split up into two different groups here of expressed genes and the unexpressed genes here. And in differential expression analysis, it's common to filter out the bottom part here, but we keep them in here in differential expression in copy number calling. And Lemma then makes a trend line here trying to fit the variance as function of the expression. And we also use the VUM with quality weights by Majewski. However, this isn't quite enough for the copy number alterations, and we, on top of this, we also want to extract the housekeeping genes. And housekeeping genes are expressed genes, so essentially it's the bulk here, but we cut out the top 2% because you often have very variable genes out in this part. It can be things like mitochondrial genes and so on that sometimes can cause a bit of trouble. So we cut out the top part to avoid that kind of outliers, and hopefully we don't lose too much power. Housekeeping genes are not only expressed, but they're also supposed to be low variance. So we're looking at genes in this area here in the lower part of this black bulb. And all the genes that are not inside there, they get a penalty in terms of increased variance. So what we get afterwards is something like this, where the housekeeping genes are kept down here. They're staying at a variance of around 0.5, while the lowly expressed genes, as well as the very top part, and the genes that have a bit on the top part of the cluster get a penalty and get moved up on the variance scale. So this is a bit, you can call this a hack, right? Because this makes us focus more on the housekeeping genes. It makes us trust these genes more, and the increased variability makes us essentially not trust the log-fold change readouts of the other genes here. So this is all fine, and this is maybe not standard, but these are like within the realm of normal differential expression. There is, however, one property of copy number calling that you don't have in normal differential expression. And that is that the signal is the expression change caused by DNA copy number. Differential expression from any other source is unwanted variance. So this is different, right? Normally, when you do a differential expression, you look for differential expression caused by all kinds of things like promoters, transcription factors, and so on. All of that is unwanted variance for us. We're only looking for the change in expression by the DNA copy number. So if we look at an example, a cartoon here, we look at genes. Each dot is a gene here across the genome. And this is the log-fold change or test sample against the reference. So we then have I draw the truth here. We have a region with 2 copies, and our log-fold change will be like spread around this line here. And we have 4 copies here, and the genes are like spread around this one slightly higher up. It's quite a large variability, as we saw from earlier. What we do know from the variance estimates is that if the variance estimates are good, then most of the log-fold changes should be within 1 error bar of the truth, which is 2 copies. And very few of the genes should be like 3 or 4 or 5 error bars away from the truth. If we find a lot of genes like that, that means that our variance estimates are too small. We're underestimating the variance. So this can be used to measure whether variance is a good, whether variance estimates that we get out from lemma and the housekeeping correction is a good description of reality. However, we don't have the truth here. We don't know whether there are 2 and 4 copies. What we do know is that each segment will have a lot of genes in it. So if we look at 2 neighbors, they will almost always be the same copy number. So we know that any 2 neighboring genes should be quite likely to have overlapping error bars, or at the very least should not be many error bars apart from each other. This has a few exceptions, right? If you're at a breakpoint when you change it from 2 to 4 copies, just that one neighbor might have a higher number. So we have to keep that in mind for later. But what we do is that we calculate we look at this thing, so we look at the difference in log-fold change, and we divide by the error estimate of the 2 log-fold changes. And the gray curve here is the expected distribution of this ratio. So we expect a lot of the neighbors to be around 1 or maybe 2 or lower. And we expect to see very few genes out in 3, 4, 5 error bars apart. So this is the gray distribution. This is the simulated expected one if the variance estimates were perfect. The blue curve here is the real data. That's the first estimates that we get from lemma boom and housekeeping correction. So we see that the blue line here is quite a lot wider. There are a lot of neighbors that are many error bars apart. That is not expected by the simulation. So what we do to correct for this, because this is a sign that we're underestimating variance. So what we do is that we add a constant to the variance of every gene. And when we do that, this blue curve will shift in towards the left, right? And we continue adding variance until the median of this distribution is matching the median of the expected distribution. And by using the median here instead of an average or something like that, we are not sensitive to outliers. We're not sensitive for like those 10 breakpoints, those 10 neighboring genes that has a real breakpoint between each other. Because there are around 20,000 genes in the genome, so the median won't be sensitive to that. And in a surprising number of cases, in almost every sample, when the medians match, the shape of the distribution also match. So this is the red boosted variance here, where we've added an appropriate constant. So this is a 1 parameter fit. And you see how well the curve fits here. And this isn't a particularly good example. Almost all samples look like this. I don't know why. I don't know why this always fits. This goes both for RNA and for exome, but for some reason, this like very simple fix is making these distributions match very well. So this isn't something where we're using the property of the copy number calling, right? We're using this neighbor property to check the variance estimate and make sure they're good. And if they're too low, we make them higher. So this is avoiding a lot of false positives, right? If we had been running with these variance estimates, we would be overly confident in our data. And it would look like lots of copy number calls everywhere. So this is how we get the log-fold changes and the variance estimates. And after that, we move on to segmentation and priority calling. But I'm not going to talk too much about that. Instead, I will go on to benchmarking and how we assess the copy number calling. And as I talked about before, we are trying to avoid simulated data. And instead, we go to TCGA. We go to the colorectal cohort and the AML cohort that have hundreds of samples each. And these cohorts have matched RNA-Seq and SNP arrays. They have exomes as well, but we don't use that at this point. So here we compare the SuperFreq calls on the RNA-Seq with ASCAT. This is a copy number caller with ASCAT calls on the SNP arrays. And plotting these, the SNP array calls in teal and the RNA-Seq calls in red, we see that both the log-fold change and BLE frequency are matching well. So it's across the genome here with the chromosome numbers. And we see this is a really good fit, right? I mean, if we didn't have to publish it and so on, have to prove it to people, like I would be happy looking at just this, right? And see, okay, this is a really good fit. It's not perfect. We see there's a bit of a mismatch here in the allele frequency, and we see some deviations here and there, but overall it's a very good fit. So we can still benchmark, right? So we see when there's, so we check when there's a gain in the SNP array, we check is that gain recalled from the RNA-Seq data. And we can then check recall for gain and loss and copy number neutral LOH, copy number neutral loss of heterozygosity. We then stratify this by the size of the segment. So we count the number of genes in the segment, and we see that when we have a segment with at least 100 genes, which is corresponding to around 12 megabases, then we have a recall of around 80%, both for gain, loss, and copy number neutral LOH. So this is quite encouraging, right? This is showing that it's perfectly possible to call copy numbers from RNA-Seq. We can do a few other assessments as well on how to measure this. For example, so here the truth was based on this SNP arrays with ASCAT. We can use a different truth as well. We can look at the most commonly mutated gene in the colorectal cohort, which is TP53. And in particular, TP53 often comes together with a loss or copy number neutral LOH, which will knock out the wild type allele of TP53 so that you get a biallelic loss of TP53. When you get that, you get an increased variant allele frequency of the TP53 variant. So we are extracting the samples that has a TP53 variant with a variant allele frequency above 70%. So this is significantly higher than 50, and it's almost a guarantee that the wild type allele is lost of TP53. This is based on variant calls from the exome. So we then expect either a copy number loss or a copy number neutral LOH. And indeed, so out of the 52 samples like this, and ASCAT is recalling either a copy number neutral LOH in green or a loss in 51 out of the 52. I think the red one here is the one exception. And SuperFreq on the RNA-Seq is doing really well as well with 50 out of 52, where it's missing the same sample here, I think, as well as having a tiny gap between the two greens here. But in the other 50 sample, we actually called something. And this is giving a slightly different number than the 80 we saw on the previous slide. It's instead giving us a 96% recall. And this is like two different assays with two different truths. And it's hard to combine the two to what to make of it. I think the conclusion is that we're doing quite well in terms of calling copy numbers. A third way to measure it is to go more to cohort level analysis as well. So this is the original paper of the TCGA colorectal data. And one of the main findings of that is that they split the samples into hypermutated and non-hypermutated based on the number of mutations called from the exomes. They then saw on the copy numbers called from the SNP arrays, they saw that the hypermutated samples have a very quiet copy number profile. There are not many copy numbers going on. However, all of the other samples, the non-hypermutated, have much more busy copy number profiles, and you also see many recurrent sites here, like chromosome 8, you see the structure 13, it's split 3 probably, you see the TP53, start of chromosome 17, and so on. We then went and tried to reproduce this analysis based on RNA-Seq alone. So we both did a somatic variant calling, even though we didn't have a matched normal for the RNA, tried to split it up in hypermutated and non-hypermutated based on that. And we also did the copy number calling from the RNA-Seq and tried to match them up in the same way as they did here. And we actually got a very similar result here. We still see that hypermutated samples are quite quiet in the copy number profile, while the non-hypermutated are much more busy, and we see the same structures here as well. So you see the chromosome 8 are the same, split 3, 13, TP53 and 17. So you see this matching up really well. So this is showing that not only on a single sample level, but also on a cohort level, we can use the RNA-Seq alone actually to reproduce the finding they used both the exomes and the SNP arrays for. So this is very encouraging, and this makes us want to take this to cohorts with RNA-Seq only that don't have the DNA analysis already done. Maybe we can do this kind of on this kind of analysis on cohorts with only RNA-Seq, where they haven't done copy number column, they haven't done point mutation column. So I'll move on from the copy numbers now and talk a little bit about the SNPs instead. And in particular, I'll go back and talk a little bit more about quality control and visualization here. So this is an AML, it's a cancer and normal sample. And on the x-axis is the variant allele frequency of the normal sample and the variant allele frequency of the cancer sample. And we see here again the heterozygous germline SNPs in the middle here. And the variants out here are only present in the cancer. So these are somatic variants out here. We can make this plot a bit more readable by adjusting point size by accuracy. So essentially we make points with a low read depth in either of the samples to make those points a bit smaller. And we now see the clusters are much more well-defined, in particular the somatic variants out here look much more cleaner. We can also separate variants that are from dbSNP, so they are more likely to be germline variants. We turn them into black crosses as opposed to the blue dots of the non-dbSNP variants. And somatic variant calling from this plot here looks trivial, right? It's just you take the variant out on the left, it's easy, and it is easy to extract those variants here. It's a little bit more difficult if you want to go down into the variants down here with low variant allele frequency and so on. It can be tricky, and this is where the different methods really separate. I think all somatic variant caller will extract like 99% of the variants up here. I'm going to show you a sample with a bit of a quality issue. So this is a sample with a MBD4 germline deficiency, and it's not cancer normal, but it's diagnosis and remission. And this is essentially cancer normal, but the normal here, the remission isn't entirely clean. So the remission sample has a little bit of cancer in it. And we see that from the variants here separating from the y-axis. The somatic variants that used to cling to the y-axis here are now moving a little bit out due to the cancer content in the normal. So this plot alone is a good way to identify this issue. You just look at the scatter plot and you see like, oh, wait, the somatic variants have self-separated a bit. It seems like we have a problem with cancer contamination in the normal. And if you run a somatic variant caller, and this depends a bit more on which method you're running, you may miss a lot of the variants depending on how picky the method is with excluding variants that are too much present in the normal. So the way to deal with that, the way that we deal with that is that we move down to clonal tracking. So what we do here is that when you perform clonal tracking on two samples, it essentially boils down to clustering the samples here. So in this case, SuperFreq identifies 3 clusters, one cluster of the heterozygous germline SNPs, and then the somatic variants here get split up into 2 different samples. And a different way to show this is the clonality, the cellular sample fraction at the diagnosis and the remission sample here. And see the red and the blue clones and the black clone growing here. Another way to show it by clone, a little bit cleaner to show it like this instead. And we're going to be more plots like this. We can then take the variants, and for example, the red clone, and we look at the mutational signature. And that means that we check what transition the SNPs are. And in this case, it's almost only C to T variants, and it's almost only C's followed by G. There are these 4 bars that dominate the signature. So we see here that the red clone here is around 200 variants total, and 80% of them are CG to TG. This is exactly what we expect from an MBD4 negative individual. We can do the same thing for the blue clone, and we find another 500 variants with an even cleaner CG to TG signature. If we do the same thing for the black clone, for the germline clone that's present both in the normal and in the cancer, we got a lot of mutations again, and we had a quite different signature. And this is quite a typical signature for germline variants. So these are not somatic variants, but these are differences between your germline and the reference genome. So how do we assess this? If we want to avoid simulations, we would like some kind of real data where we have some kind of truth, where we can check this clonal tracking, but it's quite hard to come by. So what we did in the end was a kind of compromise where we took a single cancer and normal. So we only had 2 samples. We used TCGA data for this. And we used in silico slicing and blending. So what that means is that we built 3 new artificial samples, however, using only real reads. And we then split up the genome in 4 different parts. And in the first part here, in sample 1, we took only the cancer reads, while sample 2 and sample 3 only got normal reads from chromosome 1 and 3. Moving on, then for chromosome 4 and 8, sample 1 only gets normal reads, while sample 2 and 3 gets half normal read and half cancer reads. So we mix the reads in that region. And in that way, by mixing in different ways, we create 4 different clones where we create a set of variants here. The variants from chromosome 1 to 3 in the original cancer sample will be present in sample 1, but not in sample 2 or 3. And then in the same way here, the variants in the different parts of the genome will behave in different way across the 3 samples. And this is then our truth for the clones. These are 4 truth clones. So it's not entirely real data, but it's entirely real reads. So this is as close as we can get to real data with the proper truth for clonal tracking. And we can then go and measure how well SuperFreq reproduces these things by taking these 3 samples here and plug it into SuperFreq and analyze it as real samples. And we then see that as long as the maximum clonality is above 50%, we recall the clone, we identify the clone in like more than 90% of cases. And we can also see how good we are at extracting correct variants. So we know that clone A, for example, should have only variants from chromosome 1 to 3. So if there are any variants from the rest of the genome, we know that those are miscalled. Those variants don't belong in that clone. And we see that that happens very rarely. And in particular, if we have a clonality above 50%, as we see here, almost all or sometimes all of the variants will be from the correct part of the genome. Especially this high precision of the mutation, we can really trust that the variants in a clone belong in that clone. Allows us to get the accurate mutational profiles, as I showed earlier. So we can use this actually to quality control our samples. And I'm going to show you another example. It's another MBD4 negative patient here. But instead of looking at the AML, we're looking at polyps, where this patient had polyps at the age of 18 and 27, then had AML with a bone marrow transplant, around 700 mutations and 95% CG to TG, as we saw earlier. And then 3 years later, age 37, that's another 3 polyps removed. And the research question here is, do the polyps have the same signature as the AML, as we saw? And we should bear in mind that the samples here are all FFPE, so there's a bit of noise going on that we have to deal with. If we just run a straight up cancer normal analysis of these, we find between 500 and 1500 mutations here, and a high-ish, but not extreme CG to TG mutations. Well, the last 3 samples, we had a very high mutation rate and a quite modest TG to TG rate. So this isn't quite a satisfying answer. Like, it seems like it's not quite the same signature if you just do a naive analysis like this. Instead, we go to clonal tracking. So we put these 7 samples into clonal tracking, and we find clones that are unique to each one of the polyps. And this is the expected behavior, because these are physically separated, extracted from different parts of the colon. So we wouldn't expect them to share mutations. We expect each somatic mutation to be separate to each polyp. So that's why we were quite surprised when we also found 2 clones that were shared between these 3 samples. Looking at the mutational signatures of these 2, we find there are a lot of mutations behaving like this. It's a low CG to TG rate. And maybe you remember that this signature here is very similar to the germline signature that we saw earlier. So actually, all of these variants present in 3 different polyps in different locations are from the bone marrow transplant. So this is the bone marrow donor introducing us a different germline, different DNA that then has ended up in the polyps and got sequenced. And we see here around 10% of the DNA in these last 3 is actually this bone marrow DNA. The clonal tracking here makes it easy to remove these variants though. We just discount these two clones from the analysis, and we instead look at the mutational signatures of these clones here. And we then find a much cleaner signature of 95% CG to TG, and we find a bit lower mutation rates in the samples from younger age, and then an increased mutation rate at the samples from older age. And we see that the AML samples with 700 in the middle fit perfectly in terms of mutation rate here. So this is telling a much cleaner story than the by sample analysis we did here. If you do this analysis by clone, we get out these here. So the 500 to 1500 goes to a smaller number, a much cleaner signature. So with this analysis here and removing the bone marrow transplant, we can confidently say that yes, this is the same signature in the polyps as in the bone marrow. So this is a good example of showing how the clonal tracking together with the mutational signature can be used to filter out noise and QC your samples. I want to show another example like this as well. And I'm going to show you the output from SuperFreq of this one. So this is a plot that comes straight out from SuperFreq, and this is a public data set of pediatric ALL. And here we have a diagnosis with a matched normal, and we then have 2 relapse time points afterwards. So we see the early clones, the dark blue one, that's the early cancer. The red is the relapse clone, and the cyan one here is the diagnosis-specific one. All of these are pretty low mutation rates. This is showing the number of mutations. It's 41, 39, 31, and there's a subclonal clone here as well that's present in around half of the cells of relapse, and then grows out in relapse 2. This is also a very small number of mutations with 61. Looking at the mutational signatures here, it's hard to see much because the mutation rates are so low. There's a bit of indels, and maybe you see a bit of CG to TG here, but the numbers are really so low. It's hard to say much in the early samples here. When we look at the later clones, so this one is specific to relapse 1, this one is specific to relapse 2, we see much higher mutation rates. And we also see a quite well-defined signature. And this is a typical mismatch repair signature associated with loss of function of mismatch repair genes. So as a DNA repair lab, this is quite interesting for us to see this sudden change in signature in the middle of the relapse. This isn't even the relapse clone, right? Or the subclone. It's subclones of the green one here. The green one is still low, but then everything below the green one, the purple and the orange, and the pink one here, all of those have much higher rates, and they have this MMR signature as well. So we then go and look at the river plot. So this is showing essentially the same thing. You see the blue early cancer clone here, see the light blue diagnosis specific, and the red clone is the relapse specific one, and then the green clone is the last low mutation rate one here. This is essentially the same as this plot here, it's only shown in a different way. And color matched to this are also the mutations assigned to each clone. And SuperFreq is annotating the variants and is trying to highlight protein changing mutations in cosmic census genes, so known cancer causing genes. And being a DNA repair lab, a nonsense mutation in MSH2 stands out because this is a known mismatch repair gene. And this is not only DNA repair gene, but it's also matching the signature we see in the later clones. We also see a loss of chromosome 2 in the earlier clone here. And if we go and look at the scatter plot, of the normal sample against the relapse sample, we see that the MSH2 variant has a close to 100% variant allele frequency. And this is showing that the copy number loss knocked out the other wild type allele, and then the nonsense mutation knocked out the remaining one. So we can take this information and go back here. We here have the low mutation rates. And then in the red clone, we get a copy number loss of one allele of MSH2. In the green clone here, the second allele of MSH2 gets knocked out with a nonsense point mutation. And after that, all the subclones after that, we see this very strong MMR signature and a much elevated mutation rate. So this is an example of not quality point control, but how you can use this fractional analysis and then tell this very nice story about how when you get the second loss of MSH2, you get this very clean signature. Yeah, so that's it. So I think I have a few more minutes. I wanted to show you one more thing. I wanted to show you how SuperFreq output actually looks, because the beauty of it isn't to looking at each part separately here, right? The nice part is to look at all of this together. So I'll just take a few minutes to show you what the SuperFreq output looks. This was a full cohort of the pediatric ALLs, and SuperFreq is then producing a plots directory for each patient. And this patient here was the one I was just showing. So if we look at mutational patterns, I was showing if we go back and show here. So these are the clone numbers, clone 1, 2, 3, 4. If you go in mutational patterns by clone. So this is the SuperFreq output. This is nothing messed up. This is what comes out by default. So we then see clone 4 here is the clone 4 and so on. So we see this is the green one, clone 5 is the last one, while the later one, orange one, is clone 6. We see the MMR signature and more mutations. And we see the later ones having this clean MMR signature. SuperFreq also shows you the patterns by sample, if you like to look at it like that instead. But in the two examples I showed you, the by clone patterns were probably more informative. I also want to show you the scatter plot that's used for quality control. So when I want to quality control a patient, I usually start here. So G is for germline, I think. D is for diagnosis. So this is the cancer normal sample. I'm going to be careful not to show you any germline variants here, because this is human sequencing data, and we are not allowed to show germline variants. So this one doesn't have any names or anything in it. So we see here, this is a very typical example here, right? We have the heterozygous variants in here, homozygous variants clean up here, and then we see a bit of somatic variants here. And there are not too many, right? Because there were not too many mutations at the diagnosis. It gets a bit more exciting if we look at the two relapse samples against each other. We don't have much more mutations here. We see some mutations that are present only in one relapse, other mutations present only in other. I think you know which mutation this is. This is the MSH2 that really stands out. And there's one QC issue here that's quite subtle, but after you've seen it a few times, it really stands out. And if you look at the part on the top right here, you see that the homozygous variants that are not quite all stuck up here, they're kind of like bleeding down a little bit down here. And this is something you should be a bit careful if you see this in your data. So does anyone want to type out in chat what you think this is? I'll give you a hint here that this patient had a bone marrow transplant between relapse 1 and relapse 2. So these variants up here are variants that are 100% in relapse 2, and then in relapse 1, and then in relapse 2 after the bone marrow transplant, they drop down a bit from 100 down to like 90, 95. So you start getting a bit of reference reads in that location. Still reads that align well to human, but without that variant that was homozygous in the first one. So this is the contaminating germline, right? So this is the donor that don't have a variant in that location. That's why it's bleeding down here. So you will see a similar effect down here as well. And actually, if you look at the last clone here, the cyan one, or that's yeah, that's the cyan. So that one might have a bit of donor germline variants in it. Did we get any comments there? So if you look at the by clone here, so if you look at the last one, the number 9, you actually see that this is it's not quite the same as the other MMR signatures, right? This looks a little bit more like a germline signature. And I think in reality, this might be a mix between MMR and donor. So I think if we were to publish this, we would probably not use the variants in this clone here. We would filter those out. And similarly, if you were to call like just sample against normal, you would risk getting quite a lot of false positives in this sample. So this is a way where we can tell a nice story with MSH2, and we can also use it to clean up the results a little bit due to the bone marrow. Yeah, I think I'll stop here and take